Hello, good afternoon. Am I visible, audible? Okay, only one person connected. Okay, let's wait for 5 minutes. If somebody else also want to join, then we'll start. Okay, let's start. Hello, everyone. Good afternoon to all who have joined. Can you see me? Okay, good afternoon. So, for those who have joined for the first time, a brief introduction about me. I'm Kajal Kachhawa, research scholar, PMRF research scholar from IIT BHU Varanasi. I will be your teaching assistant for this course, the host pathogen interaction, which is being conducted from January to April 2025. So I'll be taking your assignment session and I'll be helping you out in solving those assignments. So this is a brief introduction about me. I have done my master's in microbiology and BSc in biotech, and currently I'm PhD fourth year student at IIT BHU Varanasi. So I would like to know about those people who have joined for the first time. So can you please quickly write your name in the chat box along with your course degree, like you are bachelor's or master's students, and the college from where you are currently studying or your city. So please write in the chat box about yourself for a brief introduction. Just write your name and your degree, like you are bachelor's or master's students, and the college from where you are currently pursuing your degree. Write in the chat box. Those people who have joined for the first time, can you please write your name and your degree in the chat box so that we can know that which course you are currently pursuing, whether you are bachelor's or master's student, and from which college you are, just for a little introduction. Vaishnavi Bundi, master's in biotech. Okay, Maharashtra. You are currently pursuing your master's or you have completed your master's? Okay, okay. I can see so many new people join today. Yes, these recording are provided on the NPTEL only, where you are getting your the weekly assignment and your weekly lecture material. There the lecture recording is provided. And it is also uploaded on the YouTube, so you can find there on the YouTube channel also. From where you are getting the weekly assignment of Himanshu sir on the NPTEL, like you must be getting some email regarding that, for like the weekly lecture material has released or your weekly assignment is available on the NPTEL. In the NPTEL, when you will open that, my screen is shared, so I can show you. Can you see my screen now? On the NPTEL, here you must be getting like week 0, the assignment and material for week 1, for week 2, and yesterday itself the week 3 material is also released on this NPTEL. So from here, the problem solving session January 2025, from here you can get the recording session. From this recording session, you will get this Excel sheet. Here you can get the YouTube link for the week 1, and here it is a Google Drive folder. From here I will be uploading the presentations, the PPT which I am showing in the class. So from here you can get the YouTube link for the recorded session for all the week from week 1 to week 12, and from in this folder, Google Drive folder, you can get the presentation. This is at same place from where you are getting your weekly assignments. I think you guys can see the same interface when you open from the email. You might be getting the link. Okay. Others are also writing your introduction, or shall we start? Okay, medical lab technology. Okay, Kolkata. So you guys are studying this immunology course for your degree requirement, or just for the knowledge you are studying it side by side? Or there is some elective course that you can take from NPTEL? Okay. So have you also registered for the certificate course, like certification, or just enroll for the course? Because for registration for certification, it's still open. So if you want to learn and receive a certificate at the end of this course duration, then you can also register for that. Have you submitted your assignments for week 1 and week 2? Okay. Because in today's session, we will be discussing some topics that will help you in solving the assignment if you have not submitted it yet. Or also, you can make another submission for that until the deadline is over. Assignment and lecture material and assignment for week 3 is also released. So, have you seen that the material or gone through some video? Okay, no problem. Okay, I think let's start. Others also want to introduce themselves and tell about their degree, college, and their name, then they can pin in the chat box. We can take it later. So for now, let's start. Those people who have joined later on, for those, a brief introduction about myself. My name is Kajal Kachhava. I'm PMRF Research Scholar currently studying at IIT BHU Varanasi, and I will be your teaching assistant for this course, Host Pathogen Interaction, for the duration January to April. And I will be taking this weekly session. So I will be taking your weekly session on every Saturday evening 3 to 5 p.m. at the same time, and you can join from the link provided. That will be same for all the weeks. And I will be helping you out and solving the weekly assignments and some other doubts also. If you have, then you can let me know. I can take that also in the upcoming session. So if some topics are not clear from the weekly lecture material, then also you can let me know. I can take the revision of that. Or if you have some doubts, then also we can take that again. So for this course, those who want to register and get a certificate, they can also register till now. Like the deadline is open. It is until the 14th of Feb. So if you want to get the certificate at the end of this course, then you can also register for that. Otherwise, if you just want to learn it for the freely for your knowledge, then also you can join this course. So this host pathogen interaction course is divided into 12 weeks. The week 1 covers the history and development of immunology and some basic introduction of immunology, like how immunology started. And week 2 material contains the immune organs, like the primary and the secondary lymphoid organs. These two topics are already, the lecture material for these two topics is already released. So you can check it out. And recently, 1 or 2 days before, the lecture material for week 3, that is the cells of immune system, is also released, and assignment for this is also available. So you can check it out later. And similarly, the course is divided into like week 4, week 5, and week 6, where we will be covering about the immune system. And in the later on part, from the week 9, 10, 11, and 12, we will be taking the pathogen interaction, that is the virus infection and the other, like this bacterial infection and fungal infection, that we will be taking in the upcoming session. So if you guys have any doubt from these 3 week courses, then you can let me know. I can make a separate video on that, and I will cover that in the next upcoming Saturday. And if you have some questions, so that also you can let me know. These are the few of the reference books that you can follow, which is available on the NPTEL syllabus. And let's take a quick revision from the last class. On the last Saturday, very few people were there. So I would like to take a short revision of that session, like what we have studied in that week 1. We have studied about the types of immunity. Immunity is like adaptive and innate immunity. Like you all must be aware of this basic terms, innate and adaptive immunity. Uh, have you guys studied this in your courses somewhere, in bachelor's or master's or anywhere? You can quickly let me know in the chat box about yes or no so that I can proceed accordingly. So just type in the chat box those people who have joined today for the first time. Please let me know that are you aware of the terms innate and adaptive immune system? If you are aware, then okay, we can proceed directly. If you are not, then we can discuss it, then move further. Okay. So you guys, this will be my way of teaching in this live session, that I will be asking that whether you are aware or not, so that you can type in the chat box quickly. If aware, then we can proceed some fast. If you are not, then we can take that also. So, okay, thank you for your response. So if you are aware of this, then okay, fine. But still, for a short, brief introduction, the innate immunity is the immunity that we have from the birth, like from the time we are born. And adaptive immunity is like that we are acquiring it after our birth. From when we are exposing to an infection, then we are acquiring some immunity. That is the adaptive immunity. And innate immunity is present by birth. So this adaptive immunity is further divided into natural and artificial immunity. So the natural immunity is like when we are naturally getting exposed to any pathogen or any foreign particle, then we are developing some immunity. That is the natural form. And for artificial, we are intentionally exposing the organism to any pathogen or any inactive form of a pathogen so that the immune system can develop. So natural is further subdivided into active and passive. Active natural is like when we are naturally getting infected from the environment with any pathogen, then we are developing some immunity. That is the active natural from the infection. Like we are getting the common cold and flu, that is the active natural. And this passive natural is we are getting it naturally, but our body is not developing this type of immunity on its own. We are getting this from our mother, like small babies getting from the milk of his or her mother. That is the passive natural immunity. And when we talk about the artificial immunity, it is also subdivided into active and passive immunity. So the active is like immunization, like we are getting vaccinated at the childhood, like everybody have taken the polio vaccine and some other vaccines. So this is the active form of artificial immunity, like we are getting immunized for this. And the passive form is we are getting some antibody transfer, like some antibody therapy, and that comes into the passive form of artificial immunity. So these are the basic subclasses of that immunity. So you can see here, this active immunity is natural from the infection. This artificial active immunity is from the vaccination. We are getting vaccinated for this. These are the active form of immunity. Like our body will develop immunity against these two things. And when we talk about passive immunity, we are getting the pre-formed antibodies that is naturally from our mother and artificially from the monoclonal antibody therapy. We are getting these pre-formed antibodies. So this comes under the passive immunity. So this is a quick revision from the last class. This is the same thing that I have discussed right now. Did you understand the small concept of active, passive, natural, and artificial immunity? Is it clear to all who have joined? Please confirm in the chat box. Is it clear or was it fast? You want me to repeat the concept or is it clear to you all? Where is the detailed video? Okay. Uh, am I audible properly now? Was my voice breaking continuously or somewhere in the middle? There might be some network issue. Okay, there might be some network issue. You can unmute yourself and stop me there if my voice is breaking continuously so that I can know. Okay, let's proceed further for some new topic. This was the revision from previous class. If you want to see the detailed video on this, then the last Saturday video is on this only. So you can check it out there, or you can ask me in the comment box. I can take it later on again. So let's start new topic today, the history and development of immunology. Today we will be discussing about some the historical milestones in the immunology, from where the immunology concept started, from how the concept of vaccination started, how people came to know what is immune system, what is immunity. So we will be studying about this history. So the famous name you have, everyone might have heard, the Edward Jenner for the vaccination. What do you know about Edward Jenner? What do you know about Edward Jenner? Okay, and how he did, you know the story behind that, like how it started, how he came to know that it can be used for vaccination, or this is the procedure for vaccination, how he came to know about that. Anybody know the story? Father of immunology, okay, fine. Hmm. There are two different diseases like smallpox and cowpox. I'll be taking the storyline from there. So So there was a farmer, he had the cow in his farm. So there were milkmaids women. Those were milking the cow, and these women were exposed to the cowpox. That is, cowpox is a disease that is observed in the animal, in the cattle, in the cow. So he noticed that during the 1700 in England, women helped on farms. Milkmaids were women who milked the cows. Some cows got a disease called cowpox. These cows were infected with the cowpox. And these milkmaids would get sores on their hand from milking the sick cows. When they used to milk the cow, they get this type of blisters on their hands from the sick cows. The good news was that the milkmaids did not get very sick when they got cowpox, because this cowpox disease is very mild. It is not like too much severely dangerous for human beings. So they were getting mild sickness. Then Edward Jenner was a doctor in England. He often saw people who were very sick with another disease called smallpox. There was also another disease for smallpox that was in humans. So some of his patients died from this disease. This made him sad. He was very sad by seeing that the smallpox disease is quite dangerous and so many people were dying because of this disease. So that Dr. Janner heard about people talking about the milkmaids. They said that milkmaids who got cowpox did not get smallpox. Like there are two different disease. One is cowpox. It happens in the cow, and other one is smallpox. It happens in the human beings. And this smallpox is very dangerous. So many people were dying there. And this cowpox is like mild disease. They were only getting some sick and recovering back. So Edward Jenner came to know that the milkmaids who got cowpox, they did not get the smallpox. So what is the relationship between that? So for this, Edward Jenner did this thing that Edward Jenner, this woman was Sarah Nelmus, a milkmaid infected with cowpox. She had these blisters, like this cow is having blisters. So when this woman was milking this cow, she was getting blisters on her body. Like she was also getting infected with the cowpox. but this was like mild disease, no, not dangerous for her. So, and there was one other boy, James Phipps. Then Edward Jenner did this, that he inoculated this cowpox pus from the Nelmus. He have taken this cowpox pus from this infected milkmaid woman and inoculated this into this small boy, that was 8-year-old boy, that his name was James Phipps. So when he got inoculated with this cowpox, this boy fall ill, but with a mild case of cowpox, only see like very lightly infected with the cowpox. Then later on, scabs were collected from a smallpox patient. Edward Jenner have taken this some inoculum from the woman infected with smallpox. Initially, what he did, he inoculated from the cowpox infected woman, then he have taken the smallpox infected woman's that pus and then he again inoculated that into this boy. Then he found that this Phipps is unaffected. Like it is not getting smallpox disease anymore. Like this woman was getting infected with the smallpox, which is very dangerous disease. It is deadly and contagious disease. But this boy, small boy, who is already inoculated with this cowpox pus, is not getting infected with the smallpox. So from here he came to know that this proved that cowpox protected him from the smallpox. Like this cowpox acted as a vaccine for the smallpox. So he was not getting infected. So this was the first successful vaccination. And this vaccine terms is also the Latin word is vecca, which means cow. So it started from the cow. That this particular blisters in the cow can infect a woman but not very severely. But and when this blister is inoculated into a small boy, it will also get infected with cowpox, but very mildly. And once he's infected with cowpox, his body is developing some changes. His body is getting adapted to this cowpox disease, or something is happening in his body. At that time, they were not aware of this immune system, innate, adaptive, antibody, anything. They just knew that his body is getting adapted or getting some change with this cowpox. Then later on the smallpox is not affecting him much. Otherwise, the smallpox is very deadly and contagious. So this was a small story of this Edward Jenner, how it started from cowpox to smallpox. I hope this story is clear to all of you. I try to explain this in very simplified way. Is it clear that how cowpox acted as a vaccine for the smallpox? And from there, the theory of vaccination started. Is it clear to others? Okay, let's move further and learn some other story about this immunology. So this was the story of Edward Jenner. The first vaccination started from this concept. The second story is for Robert Koch. He gave the Koch postulates. What are these Koch postulates? Uh, what he tried to do, that he wanted to prove that if we are getting ill, if we are getting disease, then there are some microbes responsible for this, there are some microbes behind this. Initially, when there were no knowledge about this infection, germs, disease, microbes, no terminologies were there. Till then people used to think that they are getting ill because of some curse from the god, like they have done some sin and God is giving them some curse. So because of the God's curse, they are getting like infected or they are getting ill. Like everybody used to think they have done some pap, so that's why they were getting ill. But this Robert Koch knew that there are some microbes responsible for causing the disease. So he studied disease like anthrax, tuberculosis, and cholera. He did a small experiment, like he have taken a healthy organism, then he isolated the red blood cells and observed under microscope. And also he cultured that on some culture media petri plate. So he found that there were no growth on this, like there were no causative agent because this was a healthy mouse. There was nothing wrong with this. Under microscope, he observed clear red blood cells, and on petri plate, he observed no culture growth. At parallel, he have taken a diseased organism. He did similar thing. He have isolated red blood cells. He have seen that under microscope. He found something other than the red blood cells was also there. He didn't know what was this, but something other than RBC was there. He suspected. So he cultured that on the Petri plate there was growth, like the microorganisms were growing on the Petri plate. So he came to know that something suspicious is there. But till then he don't know what are these germs or some microbes or anything. But he knew something is there. So when he injected this with the cultured agent, this died. This mouse died. Like he have grown some culture here. He have taken this culture and injected this into a healthy mouse. This mouse died. It means something was isolated from this diseased organism, which he have seen under microscope. He have cultured on this, and then he have re-inoculated this into this mouse. This mouse died because of this. And when he again isolated this, like from here also he have isolated this on the Petri plate. So from this dead mouse also, when he isolated, then he found that the causative agent is present. Like same causative agent which was present here, it is also present here. So from here we can see that this organism have some infection that is isolated here. It is inoculated in this, and again when isolating, this is the same organism. So from here Robert Koch have given the Koch postulate, which is like the suspected causative agent must be absent from all healthy organisms but present in all diseased organism. This is what he have observed here. Somebody want to speak something? Is everything okay? Okay. So first postulate was this, that the suspected causative organism should be absent from the healthy organism and it should be present in the diseased organism. This was the first postulate. The second postulate is that the causative agent must be isolated from the diseased organism and grown in pure culture. That the causative agent must be isolated from the diseased organism and grown in pure culture. That is the second postulate. The third postulate is that the cultured agent must cause the same disease when inoculated into healthy susceptible organism. Like this cultured organism must be, when inoculated into this, it should cause the same disease. So this is also fulfilling here. The fourth postulate was that the same causative agent must be then re-isolated from the inoculated diseased organism. So from this mouse, when he have isolated it, he found that it is the same causative agent which was initially coming from here, which was isolated from here. Like some microbe is passing on from here to here, which is causing a disease. So he wanted to prove that there is some that specific microbes cause specific disease. So these were the Koch postulate. So from this, he have given these four postulates. Is it clear to all? The story of Robert Koch and his four postulates. Are you guys following or is it too fast? If I'm explaining too fast and you are unable to follow, please let me know. Please unmute yourself and stop me in between because I will not get to know that whether you are following, listening, or nobody's there. I'm speaking alone. So please let me know. So, what you understood from the Koch postulate, can anyone summarize in short, like four postulates, how it started? Mm-hmm. Yes. Yes. And it should be same at all the steps, and it should cause the same disease and same type of effect in at all the stages. It means the same pathogen is following from one step to another. So there is a specific pathogen or specific agent. At that time, they were not aware of these terms, pathogen, antigen, anything. They knew that was some causative agent responsible for causing some particular effect. So this was the Koch postulate. And let's take the third story of Louis Pasteur. He have done the first modern controlled experiment in immunology. So what he have done, Louis Pasteur successfully prevents rabies through post-exposure vaccination. My screen is visible to you guys, no? Before this, it was visible. Since you have seen those stories, Edward Jenner and Koch postulate that I have just explained, at that time the screen was visible. And you must have stopped me in between now. Then what you followed? Just listening and following. No, no, no. I was sharing the screen and showing you the pictures and images and everything. I thought you are following from there. Okay, no problem. You understood that. I will show you that again. Is it visible now? Okay, like you were just listening and understanding. Were you able to understand? For others? Very good, then there is no need for PPT if you can understand that just by talking. Then why I spend 4 5 hours and prepare this PPT? Okay, no issue. So I'll show you again what I have just explained you. I have explained you the Edward Jenner story, the first ever vaccination. This was he have developed a vaccination from the cowpox and developed for the smallpox. So this was the story I was telling you. You guys have not seen this image? Okay. Okay, I'll take a quick summary. Now you know the concept of this. I will just take a quick revision of this both two stories. It will be better to understand from this. So, in England, there were women who were milking this cow, and these cows were infected with cowpox. Cowpox is a disease that is getting infected in the cows. And this woman, when taking the milk of this cow, these women hand were also getting infected with this cowpox. But this cowpox was like very, it was not too much severe disease. So there was also some other disease like smallpox. This Edward Jenner was a doctor there. He often saw the people who were very sick with another disease called smallpox. And the smallpox disease was very dangerous and very deadly and contagious disease. So, and Edward Jenner noticed one thing that the milkmaid woman who got cowpox did not get the smallpox. So he observed this that the milkmaid who had got cowpox, a mild disease from cow, never got smallpox again. So why was this happening? So for this, he have done a small experiment. Like he have taken these cow who were infected with this cowpox. There were blisters formed because of the cowpox, and this woman, when they were milking these cow, her hands were also getting infected with this cowpox. Somebody there. So when this woman were milking these cow, her hands were also getting infected with this cowpox. So Edward Jenner There was a small boy named James Phipps. Edward Jenner taken this pus from this infected woman from the cow pox, and he inoculated into hand of this small boy, 8 years old boy. Then he saw that this Phipps fell ill with a mild case of cowpox. This boy developed cowpox infection and he fell ill. But this was very mild, not like very dangerous. And he taken another woman who was infected with the smallpox patient. He have taken the scab or the pus from here. Then he inoculated this into this boy, where he have already this inoculated cowpox that pus. So then he noticed that Phipps is inoculated with the scab of cow smallpox. Then after some days he noticed that this boy is unaffected. He's not getting the smallpox disease anymore, which is otherwise very much like this contagious and deadly. So from here he noticed that something was getting transferred from the blisters of this cow to this woman, and this thing when inoculated to the small boy, he got the illness, he got the disease. And again, when this same boy was getting infected with the smallpox disease, he does not get the infection. He is perfectly fine because the cowpox is very mild and harmless disease, and the smallpox is very deadly and contagious. So from here he came to know that the cowpox protected him from the smallpox. So this something was coming from this cow, infected cow, that was acting as vaccine for the smallpox. So this was the story of Edward Jenner. Have you understood it better now by seeing the image? Okay, okay. And the second story was this of the Robert Koch, the Koch postulate. He have taken this was the healthy organism from which he have isolated the red blood cells, and he observed that there was no growth on petri plate. And at the same time, he have taken a diseased organism. He have observed under the microscope there were some suspicious agents, some green colored cells other than the RBC under the microscope, and he cultured this on petri plate. Then he found some growth was there. Then he taken this particular culture from here and he injected this culture into this healthy mouse. Then after some time he have seen that this mouse is dying. Then he isolated from this healthy mouse again. Then he found that he got the same type of pure culture from this mouse. So, like this is getting transferred from here. Something was there in this diseased mouse, which he observed under microscope. The same thing he cultivated or cultured on a petri plate. The same thing he have transferred to healthy mouse, and same thing he have again re-isolated from this diseased mouse. So something was there. At that time, he don't know these are like pathogen microbes or what is there. He found it as a suspected agent. So he wanted to prove that specific microbes cause specific disease. So from this theory, he have given the four postulates. From here, first stage, he have given that this suspected causative agent is absent from healthy and is present in the diseased organism. And from this stage, he have given the second postulate, that is the causative agent is isolated from the diseased organism and grown in pure culture. And from this stage, he have given the third postulate, that is the cultured agent must cause the same disease in the healthy individual. And this fourth postulate he have given from here, like when this particular mouse died, he have isolated the same organism from this. So he have given this four postulates from here. So these are the four postulates that you have also seen in the class lectures. Is it clear better now? Okay. Okay. And the third story is like Louis Pasteur. Louis Pasteur successfully prevents someone is there. Okay. Just a minute. So Louis Pasteur have also done similar experiment. Sorry. He have done the first modern controlled experiment in immunology. Give me a second. Louis Pasteur have also done similar experiment. He have taken some old culture of cholera bacteria, and he have inoculated this particular this old culture in healthy chicken, and after few days he found that this chicken is free from cholera. Although this was the culture of cholera, so it is supposed to induce cholera disease in this particular chicken, but this is free from this infection. Then he have taken some fresh culture of cholera bacteria. He have taken like this previously used chicken which is not getting any infection, and he have infected the same old chicken with a fresh culture. Then he found that this chicken is still free from this disease, he's not getting infection, but he had he needed some more chicken, so he have taken this normal healthy who was not initially infected with this old culture. So when he have taken this normal healthy chicken and infected this with this fresh culture of cholera, he found that this chicken is dead because of the cholera. So why this happened? Anybody can get from the story what is happening here when he's infecting this healthy chicken with the old culture of this cholera bacteria. Then this chicken is free. And when he is infecting this used chicken, which is already done, used for an experiment for infection with cholera, and he is infecting this with a fresh culture of cholera, still it is not getting the infection. On the same time, when he's taking a normal healthy chicken, which is getting first time infection from this fresh culture, he's getting death, he's dying. So, what is happening here in the story? Mhm. Okay, so see what's happening here. This weakened bacteria can stimulate immunity without causing disease. Like this is the cholera bacteria culture. It is supposed to cause the disease, but this is very old culture. It was kept for a quite long time. He went on a vacation while keeping his culture outside. So when he returned back from his vacation, this culture was very old. So when he infected this healthy chicken, this culture was very weakened so that it can give a immunity to this chicken without causing any infection. So this chicken did not get the infection, but it got some immunity because his body developed some immunity against this particular pathogen. So when he grown a fresh culture, which is now dangerous and like it is infectious because it is the fresh culture. It is very strong. It is not weakened bacteria. So when he infected this particular chicken with this fresh culture, this chicken is have developed immunity because he was previously infected with this old culture. So because of the immunity, when he received the new culture infection, his body him itself fight from that infection and it did not get any disease. But at the same time, when he infected this with the fresh or healthy chicken, it died directly because it did not had any previous immunity because of that old culture. So this is what exactly happening in vaccines. So the exposure to weakened pathogens protect against future infection. This experiment led to the development of vaccines for disease like rabies and anthrax. So this was the Louis Pasteur experiment, from where he found that when the culture is kept for a quite longer time, it will get weakened. So from here, the concept of that we can infect the individual with a weakened bacteria, so that body itself will start forming antibodies against that. So for that, this concept started from here. Are these three stories clear to all of you? The Edward Jenner, Louis Pasteur, and Robert Koch. There were three milestone major experiments in the history of immunology. So, is it clear to all of you? Or if you have any doubt, anybody want to ask anything, or if you want me to repeat any concept again, if it is not clear, then please let me know. Because these are the milestones of immunology, these three stories. Is it clear? Is it clear to others as well? Are these three stories clear to all of you? Because there are some questions in your week one assignment from these. I have particularly chosen only those topics from where you are having a week one or week two assignments. So I'm trying to give you a short summary or a revision of those topics so that you can solve the assignments easily. If you have any doubt, then you can ask me. Otherwise, we can proceed to some other stories of immunology. Okay, okay. So those three were the milestone stories of immunology. There are other scientists as well who have done work in immunology. So one is the phagocytic theory of immunity. It was given by Eli Machnikov. So what statement he have given was that there is only one constant element in immunity, whether innate or acquired, and that is phagocytosis. The extension and importance of this factor can no longer be denied. So he said that the immunity is developing because of the phagocytosis of some cells or something. Whether it's innate immune system or it's acquired immune system, there is phagocytosis that is responsible for causing the immunity. So the theory of phagocytosis was given by Eli Machnikov. And there is one more experiment from this Emil von Behring. He have given he have said that serum act as the protection matrix. So there is another beautiful experiment in which we can see there are experiment A, B, and C. From this experiment, is everything okay? Or anybody is speaking something? So, from the experiment of Emil von Behring, what we understood that he have taken one guinea pig. This guinea pig, he have isolated some serum from this guinea pig, and the serum from this guinea pig is unexposed to any type of toxin or bacteria or anything. This is normal, healthy guinea pig one. He have isolated serum and injected into guinea pig number two. Like serum from this unexposed, uninfected, normal, healthy guinea pig is infected into guinea pig number two. At the same time, in parallel experiment B, what he have done that he inject sublethal dose of toxin from bacterial strain A. Sublethal means it is not lethal. It will not cause death in this organism, but it is somewhat toxic to this. So he have injected this sublethal dose of toxin from bacterial strain A to this guinea pig 1. Then after some time, he have isolated the serum from this animal exposed to toxin A and inoculated this into this particular guinea pig number 2. Are you understanding what is happening here? Injecting this guinea pig number 1 with sublethal dose of some particular toxin A, and then after some time he is again isolating serum from this guinea pig number 1 and injecting this serum into guinea pig number 2. Parallelly, he have done that in experiment C, he have done similar thing. But here he have injected sublethal dose of toxin from bacterial strain B. Here it was bacterial strain A, here it is some other bacterial strain, that is bacterial strain B. Then again he is isolating the serum from this animal and injecting serum into this guinea pig number 2. And after this, at this stage, this particular guinea pig number 2 from experiment A is completely unexposed. There is no pathogen, no toxin, no bacterial strain. It is only getting the serum from normal healthy guinea pig. In experiment number B, this second guinea pig is getting the serum from guinea pig number A, guinea pig number 1, which is exposed to toxin A. And this in experiment number C, this guinea pig number 2 is getting toxin from the bacterial strain B. After at this stage, what he have done, he injected all animals with a lethal dose of toxin from bacterial strain A. Like here it was sublethal dose that it might get some infection or its body might respond in some way, but it will not die. At this final stage, he have injected the lethal dose of toxin from bacterial strain A. This particular guinea pig had no exposure to any kind of strain or any toxin. This experiment B1 guinea pig had exposed to toxin A and this was exposed to toxin B. And here they all are getting toxin from bacterial strain A. So what is happening here? This particular guinea pig is dying on the spot, and this also who had initially exposed to toxin B is also dying. But the guinea pig who is initially exposed to toxin A and the sublethal dose of this bacterial strain A and developed toxin A, and currently it is getting exposed to lethal dose of strain A, but still it is not dying here even after getting the lethal dose of this bacterial strain A. So can you understand that what is happening in all these three animals? What is the difference here? Why this particular experiment B guinea pig is surviving whereas these both are getting died? Can you understand from this experiment what is happening here? Nobody would like to answer what is happening here. Did you understand the experiment? What is happening there? All of you, did you understand what is exactly what we are doing in the experiment? Like experiment A mouse is not getting anything. It is not getting any kind of bacterial strain. So it is not developing any kind of toxin. And in experiment B, we are exposing it with this particular experiment. Do you want me to repeat? Okay. So see what is happening here. Like there was a scientist, Emil von Behring. What he have done, he have taken like three set of guinea pigs. This left panel is for experiment A. It is a normal healthy guinea pig. It is a guinea pig one. Nothing is given to this. It is not infected. It is not taken any bacterial strain. Nothing is there. So after some days, this particular scientist, Emil von Behring, have isolated serum from this animal, guinea pig 1, and injected this serum into guinea pig 2. It is healthy. The healthy serum is getting injected into a second healthy guinea pig. And in experiment B, what is happening here? He have injected some lethal dose of toxin from bacterial strain A. Like this first guinea pig is already getting infected with bacterial strain A. He have got infection from bacterial A. So his body is developing toxins with respect to this strain A. Its body is developing toxin A. So when he's isolating the serum from this guinea pig, he's also, so this guinea pig is infected with bacterial strain A, its body is developing the toxin A. When the serum is isolated from this guinea pig and injected into a second guinea pig, so the second guinea pig is getting serum containing toxin A. So it is getting infected with toxin A. And in experiment C, he is doing same thing, but the bacterial strain is B. Here it was bacterial strain A, and the toxin produced was toxin A, and the injected serum was containing toxin A. In the parallel case, what happened here is he have injected this guinea pig with bacterial strain B. So this guinea pig is developing toxin B, and the serum isolated and injected into this second guinea pig is toxin B. Like toxin B is getting like from this guinea pig to this guinea pig. And in this case, toxin A is getting injected from this guinea pig to this. At this stage, what he have done, he have injected all the animals with a lethal dose. Here it was sublethal. Only mild symptoms can be seen, or body can respond in some uh, smaller way, not like very dangerous. It was sublethal. At this stage, what he have done, he have injected all these three animals. The one which is getting a healthy serum, the one which is getting toxin A containing serum, the one which is getting toxin B containing serum. Now he is injecting all animals with a lethal dose of toxin from bacterial strain A. So this have no toxin in this. This guinea pig have no toxin in this because it is healthy. This guinea pig have developed some immunity or some response with corresponding to toxin A. This guinea pig have developed some immunity with respect to toxin B. But here it is getting infected with toxin A. The strain A is getting infected at this stage. So this particular is dying because it did not have any kind of immunity to any kind of toxin, whether A, whether B, nothing is there. It was completely healthy. So it is dying. And in this case, its serum contain toxin A. Body is already started producing toxin A. So it is ready to fight against this bacterial strain A. So it is not getting infected. This particular guinea pig was initially injected with toxin B. Its body have developed immunity against toxin B. But here we are giving it the toxin A. So it is also dying. So what is happening here with the toxin A? Like this is getting injected with toxin A and it is surviving. It is not getting diphtheria infection. Whereas these both are getting infection. So can you understand what is happening here? Is this experiment clear like what we are doing here? Is the procedure clear? Okay. So now can you understand what exactly happened at that case? Why two are two guinea pig are dying whereas one guinea pig is getting no infection? So now anyone can summarize what is happening or what you understood from that experiment. Anybody? Anybody want to summarize the experiment? What exactly we have done there in the experiment? Okay. Means experiment is clear, no? And second one? Okay. Means his body is ready to fight against the strain A, not against the strain B. Is this experiment clear to all of you? If you have any doubt, then I can again repeat, no issue with that. You can feel free to ask me if any doubt is there or anything is confusing. You can let me know. See, my motive is that you understand few little small stories of immunology and you feel very happy by learning the immunology. Like I used to feel happy. So there is no point of getting stressed out and just mugging up the stories like A is given, B is given, C is dying, what is happening. No need to mug up those things. So if it is not clear, you can discuss with me or I can repeat it that again. So please feel free. If it is clear, then please confirm in the chat box so that we can proceed. Okay, for others also, is it clear? Okay. So for a summary, what is happening here? In experiment A shows that without immune protection, diphtheria is fatal because there is no immune protection in this experiment A. No bacterial strain exposure, no toxin development, no immune protection. In experiment B demonstrate that immunity can be transferred through antibodies in serum, that is passive immunity. This guinea pig was exposed to bacterial strain A and it developed serum for this toxin A, and these antibodies are transferred to this guinea pig. So it can be like immune antibodies can be transferred from one organism to other. In it is called passive immunity. And in experiment C proves that antibodies are specific to the strain they were produced against. Like this guinea pig. What happened? Please unmute yourself. Okay. The experiment C proves that antibodies are specific to the strain because this guinea pig have developed antibodies to the toxin B. And when it is getting exposed to toxin A, its body is not ready to fight against that. So it is getting dead here. So this experiment confirmed the role of antibodies in immunity and laid the foundation for passive immunization using antitoxins, that is the antibody therapy, and vaccine development against bacterial diseases like diphtheria and tetanus. And the Nobel Prize in Medicine was awarded to Emil von Behring for this work in serum therapy. So Emil von Behring have suggested that serum act as a protection matrix. There is another small story for toxin neutralization assay from Paul Ehrlich. So what is happening here? When this particular cell is getting infected with the toxin molecules, the cell is getting damaged. Okay, this is the fine. As we can know, if there is a cell, it is getting interaction with any toxin molecule, the cell will get damaged. But if we give any neutralizing toxin, like toxin is there, cell is there. If we give any antitoxin, this antitoxin can bind to this toxin. So this toxin and antitoxin will form a complex and this will toxin will get neutralized. So now cell will not get damaged because the toxin is not freely available to go and interact with the cell because the antitoxin is forming a complex with that and neutralizing this. So this was the toxin neutralization assay by Paul Ehrlich. So cell will not get damaged. So what is the need for this experiment? What is happening here? See, these antitoxins are antibodies that neutralize toxins before they cause harm. Used in passive immunity, where pre-made antibodies are injected into a patient for immediate protection. Like, you know what happens in snake bite, snake venom, what happens there, what we do when we have any bite with the snake, any snake bite. Mhm. So, what is that anti-venom there? This anti-venom is the antitoxin that is going and binding to the snake toxin. So, that toxin will not cause harm to our body cells. The same concept is here. Like, just suppose this is the snake toxin, the venom from the snake, and we give anti-venom. This venom will go and bind to this snake venom. It will bind and form a complex, it will get neutralized. And our body cells will not get damaged. So this is what we exactly do when we give the antitoxin. So few examples of antitoxin are in diphtheria, we give diphtheria antitoxin. In tetanus, we give the tetanus antitoxin to prevent muscle paralysis. In snake bite, we give antivenom works on the same principle. So the antitoxins provide immediate protection in cases of severe bacterial toxin infection, and it forms the basis for passive immunization used in emergency treatments, and it led to the development of vaccines that stimulate the body's own protection of antibodies for long-term immunity. Are all these experiments clear to everybody? All the experiments are clear, or you want anything to repeat? For others as well, are all these experiments clear? Then we will be moving further to other experiments. There are so many experiments in immunology. Okay. Okay, so there is one more that this fever phenomena. What he said was this. The fever phenomena was discovered by Richard Fever in 1894. It describes the bacteriolysis, that is the destruction of bacteria due to the immune system, specifically through the action of antibodies and complement proteins. This discovery was fundamental in understanding humoral immunity and the role of antibodies in bacterial clearance. So, what is happening in this particular phenomena is that he have injected Vibrio cholerae into normal animals. Like these animals were normal. He have injected Vibrio cholerae bacteria. So when Vibrio cholerae, that is the cholera causing bacteria, was injected into the peritoneal cavity of non-immune, that is the normal animals, the bacteria multiplied rapidly, causing infection. Parallelly, when he injected this Vibrio cholera into immunized animals, here the animal were non-immunized. They have not received any type of like antibody or anything before that. These animals were completely normal. That was the like first exposure to this Vibrio cholera. Then in animals that had previously recovered from cholera infection, the injected bacteria swelled up, dissolved, and disappeared within 20 minutes. This bacterial destruction occurred without any direct cellular activity. That is, no phagocytosis was there. Just a minute. Sorry for my bad throat. So he have injected this into the immunized animals, and this bacteria disappeared within 20 minutes. Then at third case, what he have done, he have taken the serum from immunized animals, destroyed bacteria in the test tubes. Like what he have done, when the serum, that is the antibody-rich fluid from the immune animal was added to a test tube containing Vibrio cholera, the bacteria were lysed, that is destroyed in the same way. This proved that bacteriolysis was due to the antibodies and complement proteins in the serum, not due to the immune cells. So what happened here, in short, he have injected this Vibrio cholera into a normal human being. Okay, in the peritoneal cavity of normal human being, he found that it Vibrio cholera was causing infection is in this particular case. In second case, what he have done, he have injected the same Vibrio cholera into the patients who have recently recovered from this cholera infection. Like those individuals have some type of immunity towards this cholera infection because they were previously infected with cholera, then they recovered, and it is the second infection. So the injected bacteria swelled up, dissolved, and disappeared within 20 minutes. Like they did not cause any kind of infection, and these bacteria were destroyed. So for in order to confirm that how this happened because of any like phagocytosis, any cellular activity or any antibodies were there, in order to confirm that he have isolated serum from these immunized animals. Like these immunized animals, he have taken serum. He have taken out the serum from these immunized animals. And when the serum from an immune animal was added to a test tube containing Vibrio cholera, here, what he was doing, he was injecting this Vibrio cholera into human being. In this third case, what he have done, there was a test tube which contained Vibrio cholera. He have isolated serum from that immunized individual and put the serum into the Vibrio cholera. So in serum, there were no cells. It is not like cellular activity. So serum contained only antibody-rich fluid. So the bacteria were lysed in the same way. Like here, how they were lysed, like these bacteria were swelled up, dissolved, and disappeared within 20 minutes. Same thing happened here. So from this, he proved that this bacteriolysis, that is the lysis of bacteria, death of bacteria, was due to the antibodies and some complement proteins present in the serum. Because here he have taken out only the serum, no cells were there. So he have proved that this is because of the antibodies and complement proteins present in the serum and not due to any type of immune cells. So this was the fever phenomena. Is it clear to all of you what happened in these three cases, what he have done, and what was the result and outcome of this? Is this experiment clear? Okay. For others, is this clear or is it confusing and I need to repeat this again? Please confirm whether it's clear or not. Are you guys following what is happening in these experiments? Okay. So, what was the main key points of this fever phenomena? It is given here. The vibrio inoculated into the peritoneal cavity of normal animals rapidly multiplied, but the same vibrio in the animal that had recovered from the infection and underwent bacteriolysis, that is, they rapidly swelled up, dissolved, and disappeared in 20 minutes. This took place without any cellular activity whatever, and in fact, the serum of the immune animal produced the same lysis in the test tube. This is the fever phenomena, which is still in use for differentiating closely allied species. So, but all this very much complex thing is explained, same thing is explained here with these three type of experiments which is conducted by the scientist. So let's move to another study, that is a bactericidal power of blood. It is from this Nuttall and Buckner. These two scientists were there. So what George Nuttall have said in his study, what was his key finding? That blood has inherent bactericidal power. So what he have done, he have taken the defibrinated blood, that is blood without fibrins, the clotting protein, was found to kill anthrax bacteria, which is Bacillus anthracis. However, when blood was heated to 55 degrees Celsius, it lost its bactericidal ability, suggesting that heat sensitive factors, that proteins, were responsible for killing bacteria. This indicated that blood contained natural antimicrobial components, which later became known as complement proteins and antimicrobial peptides. So what is happening here? He have taken defibrinated blood, that is blood without the clotting factor, and it was able to kill the anthrax bacteria. But when he have heated this blood up to 55 degrees Celsius, it was not able to kill the anthrax bacteria, because what happened while heating the blood at 55 degrees Celsius, that it lost its bactericidal activity because of the heat sensitive factors, those were denatured. So this suggested that there are some natural antimicrobial components present in the blood. These are now known as complement proteins and antimicrobial peptides. So this study provided early evidence that blood alone without immune cells could destroy bacteria. Like there was a debate that whether the immune cells are responsible for fighting from the infection or the antibodies present in the blood. So these scientists have done separate studies in order to give their opinion on whether these immune cells are responsible or this blood is responsible. So in this last study, the scientists, the fever, have told that these antibodies are responsible for the bacteriolysis, but not the immune cells. And this also, this scientist George Nuttall is trying to tell that this blood alone, that is without any immune cells, could destroy bacteria. And this paved the way for research into antimicrobial substances in body fluid, and it suggested the presence of heat sensitive proteins which are responsible in providing immunity. Those are now known as complement proteins. So this was a short study which tell that blood has inherent bactericidal power, because when he heated this blood up to 55 degrees Celsius, this bactericidal power was lost. So this is the story of George Nuttall. Is it clear? Okay. There is another similar study from Edward Buckner. What he told, that he identified a bacterial killing factor in serum known as alexin. So Buckner discovered that blood serum, the fluid portion of the blood without cells or clotting factor, could destroy bacteria. He named this factor alexin, which later known as complement system. The similar story as we have seen in this case, George Nuttall case, it is also similar study from Edward Buckner. The complement system consists of proteins that enhance immune responses, helping antibodies and phagocytes destroy bacteria and infected cells. So, from that study, he confirmed that immunity could be mediated by soluble factors in blood. That is the humoral immunity, that is consisting of antibody and complement proteins. And it led to the discovery of the complement system, critical part of the immune response. So, this is also a similar study where he wanted to show that the blood serum can destroy bacteria, and he called this factor as alexin. Later on, this is known as the complement system. So this is also similar study from this two scientists. So these both scientists have given the this bactericidal power of blood. Josh Nuttall have used this blood, and when he heated up to 55 degrees Celsius, this bactericidal power was lost against Bacillus anthrax bacilli. And Buckner have also done similar study where he told that this particular factor alexin could destroy bacteria. And these both have suggested some way to for the complement system, complement proteins that we will study in separate unit. So is this study also clear to you? George Nuttall and Edward Buckner. Is it clear? Okay. There is another story for bacterial agglutination from this Gruber and Durham, and later on joined by this Vidal. So what they have told that Max von Gruber and Herbert Durham have told that bacterial agglutination, it is a process where bacteria clump together in the presence of specific antibodies in a serum sample. It occurs only when the antibodies in the serum are specific to the bacteria, making it an important tool for identifying bacterial infection. Like in short, what they are saying, when we have some bacteria in the blood and we have some specific antibody in the blood, then these both will interact with each other and it will cause agglutination in that. So, like when serum are specific to the bacteria, making it an important tool for identifying bacterial infection. What type of bacteria are present that will interact with that particular type of antibody only. So, this discovery by Max von Gruber and Herbert Durham found that when bacteria were mixed with immune serum, they clumped together. That is, they caused agglutination. They demonstrated that this reaction was highly specific. Each antibody only agglutinated its corresponding bacterial strain, and this proved that antibodies in the blood could recognize and bind specific bacteria. So they have given this concept that each antibody can bind to the respective bacteria and can cause agglutination. This concept was further used by this scientist, George Fernand Isidore Vidal. What he have given, he said that bacterial agglutination for typhoid fever diagnosis. So he have used this concept that each antibody only agglutinated its corresponding bacterial strain. He have used this concept for the development of this Vidal test, which is known as the Gruber-Vidal test or the Vidal reaction that we get done for this typhoid fever. Have you heard this name, no? Vidal test. It is very quite commonly used, the Vidal test. Are you aware of this name, Vidal test? Okay. Whenever we fall sick or we have any fever, then we go to any doctor, then he suggests us to get a Vidal test that done. That is just a test using our blood. So it is used for the diagnosis of typhoid because typhoid is a very common fever. It is caused by Salmonella typhi. So okay, sorry for that. Vidal test is used for the diagnosis of Salmonella typhi that is causing the typhoid fever. So see what is happening here. So how Vidal test work? When a patient's serum that containing antibody is mixed with Salmonella typhi bacteria, that is, it causes the typhoid fever. So when this patient's serum that might be containing some antibodies, because if you have typhoid, then your body is producing antibodies against that. So if your serum is mixed with Salmonella typhi bacteria, then if the patient has typhoid fever, their serum will contain antibodies against Salmonella typhi, and this bacteria will clump together, cause agglutination, and confirm the infection. So it is a very quite common blood test. So it is the first serological test to diagnose bacterial infection. So how this came into this knowledge, that these two scientists, Max von Gruber and Herbert Durham, have done some studies and they told that bacteria clump together in the presence of specific antibodies. And each antibody only agglutinate its corresponding bacterial strain. And this Isidore Vidal has used this their concept and he developed this Vidal test. So Vidal test is just like that. Have you seen the blood test, blood group test? Have you seen those slides or studied about that? How we get to know what is our blood group, like whether it's blood group A, blood group B, blood group O, blood group AB, whether it's positive or negative, how we get to know by the blood group test that is working in the same way that when an antigen and antibody is interacting with each other, they will form agglutination. They will form clumping in that, yes, by using antisera. So the antisera here is what? That is the antibody and the antigen that is present in the blood. So from that we are getting which blood group is there. Similarly, it is for Vidal test is for Salmonella typhi bacteria that is acting as an antigen and the corresponding antibodies which are isolated from the patient's serum. Because when we are getting infected with Salmonella typhi, our body started producing that antibody against Salmonella typhi. So, is Vidal Gruber Vidal test clear to all of you guys? Okay. So these were the few interesting stories of immunology, and there is one slide in the presentation where few scientists are supporting the cell, where few scientists are cellularists. That is, they are supporting the this theory that chief defense of the body against infection resided in the phagocytic and digestive power of the macrophages. Like they are saying, immune cells are responsible for providing defense from the infection. But there are some scientists, those are humorist, they are telling that only the soluble substance of the blood and other body fluids could immobilize and destroy invading pathogens. Like they are saying, there are some soluble substance like antibodies, some complement protein, uh, they are found in the blood. They are helping us to prevent from the infection. So there are some humolarist and there are some cellularist. So when we will study this immune system in detail, like the cells and organs of immune system and cellular and humoral theory of immune system in the upcoming session, then all these theories will be like everything is okay. Some are saying only the soluble factors are responsible, some are saying only the cells are responsible. But from the current immunology knowledge, these both are working parallelly in protection from the infection. So there are some theories, there are some scientists. This you can find from the presentation or weekly material. Most of them we have covered today, but still, if you want to discuss any theory in detail, we can take it. So there is a next topic. So till here, are all the theories clear to all of you, or you want a quick summary of each and every theory? Because it is the history and development of immunology, how it actually started. Till here, is everything clear to all of you? Is everything clear till here, or others? Okay, okay. If you have any doubt, any confusion, or any curiosity, or anything, you can ask. Okay, okay. Thank you everyone for your response. I will show you what we have studied today so that you can take a quick recap of everything. And if you need to discuss anything, let me know. We will stop there. So here in this slide, we have studied types of immunity, adaptive and innate immunity and their subtypes, that is natural and artificial. And again, their subtypes, that is active and passive. We have studied what are these active and passive, what are natural and artificial, what are innate and adaptive, how we are getting this immunity. It was a quick revision from the previous Saturday class. So we have seen this type of immunity here and the examples. From here, we came to history and development of immunology, where we have studied the first classic example or the classic story or the milestone of immunology, that was the Edward Jenner story. It was for this cowpox and smallpox. He have developed a vaccine for smallpox by using cowpox serum. So he have done this particular experiment and he came to know that cowpox protected him from smallpox. The cowpox is acting as a vaccine for the smallpox. So from here, the story of vaccination started. Then from here, we came to this Robert Koch experiment, that is the Koch postulates. He have given this four postulates. You should remember all the four postulates, but please don't mug up these postulates. Just remember this diagram in your head. Just imagine this diagram and start from here. Like from healthy, nothing is grown, from diseased, something is grown. When this something is injected into some organism, it is getting response, and when something is isolated, it was the same thing that was initially there. So just remember all these four postulates by this story. Please don't mug up, otherwise you will forget. The next was Louis Pasteur's story. He conducted the first modern controlled experiment in immunology. He have done the experiment on chickens from healthy, and he have taken this infect this already like this injected or infected chicken but did not develop any disease. Then he injected again with the fresh culture, then also it was free from disease. And we have seen this particular story of this cholera for the Louis Pasteur. Then we have seen the phagocytic theory of immunity from Ali Machnikov. Then we have seen the serum as protection matrix experiment from Emil von Behring, where he have taken three different this guinea pigs and injected with different toxin, and then he found that this toxin A containing guinea pig have developed immunity against this toxin A or the strain A. So from here we went to the toxin neutralization assay by Paul Ehrlich, where we have seen how this toxin and antitoxin are forming a conjugate, and it is protecting our cell from the damage. So this is actually what happening in the case of the snake bite, tetanus, and diphtheria. From there, we have seen this fever phenomena. Fever was that this Vibrio cholerae phenomena. When injected into normal animals, it caused infection. When injected into immunized individuals who have recently recovered, there was no infection. From here, he has said that bacteriolysis was due to antibodies and complement protein in the serum, not due to the immune cells. From there, we have seen the bactericidal power of blood, where we have seen this George Nuttall and Edward Buckner experiment, where they told us in blood there is a complement protein present that is protecting us from the infection. From there, we have seen this Gruber and Durham experiment. Their theory was that bacteria clump together in the presence of specific antibodies in a serum sample, and each antibody only agglutinated its corresponding bacterial strain. And from using their concept, Isidore Vidal have given this Vidal test, which is commonly used for the this detection of Salmonella typhi infection. From here, we have seen the cellularist and humolarist theories. So, till here, is everything clear to all of you, or you want to summarize something, or ask something, or any suggestions, or any questions? Everything is welcome. Please feel free to speak up. How was today's class? Did you learn anything new, or everything you already knew, or every concept was already clear to you, or you have heard something for the very first time, or you have already heard of these things, but these were not very much clear, and today are they clear? Is there any suggestion or any comment from your side? These topics I have covered with respect to what questions are asked in your week one assignment, so that this all the history and development of immunology topics are clear for you, so that you can solve your weekly assignment. It was with respect to that. Any suggestion, any comment? Okay, thank you very much for the response. Anybody else also have any suggestion or any comment or anything you want me to improve for the next session? Like if my speed is fast, or my vocab is not clear, or my way of explaining thing is not clear, or anything, any suggestion is most welcome. Because I'm also student, I'm also learning. So we are learning together. So any type of suggestion is most welcome. Anybody, any suggestion, any comment, or any specific topic you uh, want me to explain in the next class? Then also you can let me know, because I'm trying to cover everything with the weekly materials that is released on the NPTEL portal. But for your request and your suggestion about that cells of immune system topic and the questions about that, I'll be taking that in the upcoming classes. when there is a class with respect to that, I will be covering it there only because the material is already released now. So you first go through that, then it will be more clearer to you. Okay. Anything other than that? Okay. Any doubt for the today's class or everything is clear? Okay. Are these things already clear with the lecture video and the PDFs provided to you? Okay, so I had few more topics for today's class, but I don't think so. We can cover it up in today's class, like some Nobel Prize winners of immunology that I think you might have seen this from the material itself. There is no more discussion in this. And for branches of immunology, also, it is like some direct definitions and concepts also that you can see from the video lectures. If not clear, if you have any doubt, then you can post it on the discussion forum, or you can put it in the chat box, or you can put a comment on my video so that I can take it from there, or you can drop me an email. No issue with that. I will take your doubts and try my best to clear that. So, branches of immunology, then I had this organs of immune system for the upcoming class. This we can take in the next class. So, for now, this was from my side. So, you can have any discussion if you want, or any suggestion for next class, what you want me to take. When the date is your that assignment? What is the last date for your assignment? The due date for submission? 5th of February. Okay, then my next class will be on the Saturday, that is 8th. Okay, then I will solve those assignments also in the next class. We'll have a discussion on that. Other than that, any topic you want to study for the next class along with the assignment? You can go through the video lectures from the NPTEL channel, then read the PDF, then we can have discussion on Saturday. Okay. Did you get the video lecture for the first class, week 1 class? The video link lecture for week 1 that is uploaded on my YouTube channel, and the link is provided in that NPTEL study material space. And this video will also be uploaded at the same space like I have shown here now in this host pathogen interaction, where you are getting the study material for every week, the assignment and the video lectures, everything you are getting here. At the bottom of this, you will be getting here this problem solving session for this January 25. This recording you can access from here so that you can get the YouTube link for this. So this is a week 1 video is uploaded here, and at same space the week 2 video will be uploaded. So you can find it, and I will also share the presentation, this PPT on that. same space. So you can get it from there. Other than that, any suggestion, any comment, anything? Okay, no problem. If you have any other suggestion, any comment, you can post it on the discussion forum. So thank you everyone for joining the class. Thank you for your suggestions and your response. We will meet on next Saturday at same time, 3 to 5 p.m. Okay. Okay. Thank you. Thank you everyone. You may leave.